Today we're going to continue with 7.1 and we are going to look at DNA sequencing and how it's done. So this is one of the applications, and to be more precise is how the Sanger method uses nucleotides containing dideoxyribonucleic acid, also known as dideoxynucleoside triphosphates, to stop DNA replication in preparation of samples for base sequencing. So this is the most widely used method for DNA sequencing involving the use of these dideoxynucleotides. So we are going to see today how they are used. So the first thing we need to understand is, what are these dideoxynucleotides? So like I said before, sometimes in your book or in your PowerPoints, you're going to see them referred as dideoxyribonucleic acid. Don't be confused, they're the same thing, okay? So these dideoxynucleotides, or DDntps, they are a form of nucleotides, but they lack the 3' hydroxyl group necessary for forming a phosphodiester bond. So as you can see here in this picture, here's an example of a dideoxy nucleoside. It lacks the 3' hydroxyl group, so it cannot form bonds and it cannot extend the DNA chain. Okay, so these DDntps, the nucleotide can vary in base. It could have an A for a base, or a T, or a G, or a C. Okay. As opposed to your regular deoxynucleoside triphosphates, this is what your regular nucleotide would look like. And this is what we call just your regular DNTP. So this is a regular deoxynucleoside triphosphate, and the nucleotide, again, can vary in the base that it has. It could be A, T, G, or C. So in other words, if you want to think of these as just your regular nucleotides, and these as your nucleotides that cannot keep elongating or forming bonds. Okay, so how are they used? So here let's look at a picture. So let's say we have a DNA sequence that looks like this, right? And we're trying to find out what the actual sequence of nucleotides is, what the actual sequence of bases is. So what we're going to do is we're going to add different DDntps that are going to further prevent the elongation of the DNA that is getting copied. So here, for example, and here's their example that they're using here. If I add DDGTP, remember I told you that these DDntps can be made up of G, C, A, or T. So if I add one that has G, then that means that every time I am copying this DNA strand, every time I stop the sequence right here, I know that the corresponding DNA sequence is going to be C. So I know that this fragment here, every time it stops, it's going to stop at a C. Again, this fragment, which is a little bit longer, every time it stops, it's going to stop at a C. Again, this fragment, every time it stops, it's going to stop at a C. And the same thing here, right? So sequence terminates when the DDntP is incorporated and the fragment lengths reflect the base position of the sequence. So by adding these DDntPs, I'm making these really short fragments, okay? So this still might not make a lot of sense until we move further here to this next picture. So these DDntPs can be used for sequencing purpose, and what we call this DNA sequencing method, we call it the Sanger method, named after Frederick Sanger, who developed this method, and it started to be used back in the 1970s. And it's an actual, it might look a little bit complicated here, but it's an actual simple process here, and it's very precise, and it's still used today. So let's say we prepare four, let me get my pen here, 4 PCR mixes, okay? And remember what PCR is. Now, PCR stands for polymerase chain reaction. So that's a way to take a DNA sample or a DNA sequence and make multiple copies of it in a very short period of time. So let's say I create or I make 4 PCR samples. So in these PCR samples, in each sample, I need to add the one particular DDntp, and remember there's 4 different types of bases. So one DDntp is going to contain thymine, the other one's going to contain adenine, the other one's going to contain guanine, and the other one's going to contain cytosine. So I'm going to have 4 different mixes with 4 different DDntps for the 4 bases. In addition, in these mixes, I'm going to have DNA polymerase, I'm probably going to add primers, and I'm also going to add just regular, normal nucleotide sequences. So I'm also going to add just my regular DNTPs, right? Because I need my sequence to copy itself with regular DNTPs as well. Okay. So now I've prepared 4 PCR mixes with what I just told you. So I'm gonna put these mixes in a PCR machine, and a typical PCR machine will generate over 1 billion DNA molecules. So that means that each PCR mix should generate all the possible terminating fragments for that particular base. So let's say I am trying to figure out the sequence of this DNA strand right here in purple. So pull this up here a little bit. So notice that the strand here is 5 to 3, so that means that I'm going to start copying in a 5 to 3 direction. So I'm going to start right here in the 5' end. So let's say I add this mix in first, right? The DDGTP mix first. So I'm actually going to, I'm actually going to start right here by adding this mix, the DDTTP mix first, okay? So I'm going to start right here. I'm still starting in the 5' direction. So anytime the DDTTP mix finds an A, it's going to stop, right? Because it's not going to continue elongating the DNA strand. So this is going to be my first fragment right here. It's going to be very short, okay? And then if I continue with the DDTTP mix, this is going to be my second fragment right here. It's going to be longer, and I'm only going to get 2 fragments for DDTTP. For DDATP, okay, I'm going to get my first fragment here. So anytime, so here I'm copying, right? And anytime I hit a T, I'm going to stop. And I'm going to copy again. I hit a T, I'm going to stop. So I have 2, oops, right here. I have 2 fragments here. I have 2 fragments there. And then right here, again, I have another fragment there. So I have 3 fragments here with DDATP. Third fragment, DDGTP. I'm going to continue copying here, and then I'm going to stop, right? And then again, I'm going to continue copying another one here, and I'm going to stop, and I'm going to continue copying another one here, and I'm going to stop. So I ended up with 3 fragments with my DDGTP mixture. And then with my DDCTP mixture, here's going to be my first fragment when C reaches G and it stops. And then my other fragment right here when my C reaches G and it stops. So that's 2 fragments. So here are all my fragments for this DNA sequence. So I have a total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 fragments. Okay? So now what does that mean? So that means that I can take these fragments and separate them in a gel, okay, and then read them according to their size. So let me erase this. So notice that this first fragment here is a very small fragment. So if you're very short, that means you're going to travel very long distances in a gel electrophoresis. So remember that the gel electrophoresis is going to run in this direction. DNA is going to be placed inside these wells here, right? And then this fragment here is going to be able to move very long distances towards the positive cathode here in the gel electrophoresis chamber. And then this fragment right here is somewhat medium length, right? So that's this fragment right here. Notice the three fragments here for A. One is, this one's the smallest. It's the one that can travel the furthest. And then this one, they're about the same size, so they're close together. And then this one is a very large fragment right here, so it can't travel very far, so it stays up here on the gel, right? Here are my three fragments for G. 1, 2, 3. This one's a very long fragment here, so it can't travel far at all. This one travels midway, and this one is a very short fragment here, and it travels kind of fast. And then we have two fragments here, right? For C, here's one fragment, and here's one fragment. Of course, this one's smaller, travels faster than this one that's larger, okay? So now I've separated all of my fragments in the gel. So how do I go about reading now my DNA sequence, right? Because remember, I don't know what this DNA sequence is. All I have are these fragments, and I'm reading them in the gel. So I've separated them. Now, luckily, we have machines that read these, right? And we don't have to like read them manually. But if I were to read them manually, what would I read? I would read the G here, okay? G, and then I would read the A, and then I would read the C, right? I would go according by the length of the fragment, and then I would read the T here, and then I would read the G here, and I would continue reading that. And I know that each fragment corresponds to a DDntp that has stopped at a particular DNA sequence. So I know that if this fragment, the first fragment that I read is G here, I know it corresponds to a C. And if this fragment is A, it corresponds to a T. And if this fragment is C, it corresponds to a G, and so on and so on. So then I end up with my whole DNA sequence that I'm able to read. But like I said, you're not responsible for learning the how to read these sequences, by no means. You're not even responsible for describing the process of the Sanger method. I think the important part here is for you to understand, like it says in the application, what these DDNTs do, okay? And DDNTs are used to determine the sequence of DNA in the Sanger method. Why? Because they cannot form bonds here, and they stop DNA replication at different fragments. So there's a few videos also that you can watch on the Sanger technique. It is, as you can tell, it's an in-depth, a bit complex way of, it's a simple technique, right? But it sounds complex, and it's definitely not as easy to understand. So let me know if you have any other further questions, and go ahead and read your book, your PowerPoint, and watch the other videos.